Okay, good day everyone. So, my topic is all about the protein synthesis in bacteria. So, protein synthesis is carried out in the cytoplasm. It begins with DNA duplication by mRNA, or it is called transcription. So, mRNA then migrates to the ribosome where tRNA transfer information from mRNA to RNA, it is called translation. So, in protein synthesis, we look at two major steps. One is a transcription and the other is translation. So, in transcription, we are going to transcribe the DNA into a message. So, in a cell of bacteria, the DNA is in a nucleus. So, therefore, we're doing kanang transcription sa nucleus. In a step of transcription, an enzyme that is called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bound together to form single-stranded mRNA. So the mRNA stands for messenger. Messenger RNA nag-consist siya og message made made of RNA that has been nagbase sa DNA. So the mRNA can go out of the nucleus into the cytoplasm where it's going to attach to the ribosome. So ribosome make protein. The ribosome made of rRNA, or it stands for ribosomal RNA. So the ribosome is going to build a protein in the next called translation. So muna siya tawag translation na. So in the cytoplasm, there are tRNA molecules available. T stands for transfer RNA, and they carry amino acid. And the amino acid is the monomer for protein, and it's a building blocks of protein. Since kanang we're making protein, we're going need those amino acid. So nanghinanglan tag amino acid para makahimo og protein. So the tRNA is going to bring those amino acid to bring those amino acid together to make up. But how? But how does tRNA know what the amino acid to bring? Mao na nga nanghinanglan ta og mRNA, or the messenger RNA, because it's going to direct which kanang tRNA come in, and therefore amino acid are transfer. So dapat kanang naay mRNA para siya mo signal unsa na information na abot nimo unsa nga tRNA nga abot para it's going to transfer sa ribosome. So there are stages of protein synthesis. It continues takes place in 3 stages. There are initiation, elongation, and termination. So when we say initiation, ang mahitabo sa initiation naay gitawag og ribosomal subunits, the large and small. So mo separate na silang duha with the help of initiator factors, the IF1 and the IF3. So mag-separate na siya. Ang small subunits mo-attach na ngadto sa shrine delgado. When we say shrine delgado, line na siya na mo consist of codons or letters. So kani siyang codons mao na nga mga letters nga dapat mag-attach sa letters or codons nga gidala sa tRNA. So kani po siyang tRNA nagdala po na siya og amino acid. So mabitaw to nga ang amino acid na adyo siya kana naa siya sa tRNA. So dapat makibaw tag unsa nga tRNA nga gidala-dala. So kanang amino acid once maghimo na og chain which is called peptidoglycan chain, mao na siya pwede na ma-synthesis into protein. So adto na ta sa ni-attach ang small ribosomal subunits sa strand delgado. So ni-attach naman ang small ribosomal subunits sa strand delgado na ay moabot nga tRNA. So kana siyang tRNA, nagdala na siya og amino acid which is the formal methionine. Mao jud na siya ang kinaunhan nga moabot ang formal methionine. So mo-attach na siya sa ngadto sa codon sa strand delgado. Pero gisigurado na siya sa strand delgado nga mag-attach na mag-match jud na ang kinaunhan. For example, ang codon sa tRNA kinaunhan kay U. Ang kinaunhan pud sa strand kay A. Match sila nga ang pikas tRNA is A nya ang strand U. So match gyapon sila. Ang ikatulo is G, so ang strand is C. By the way, the tRNA consists only of 3 subunits codon. 3 ra jud na sila ka units. So 3 ra jud na sila ka letter na mo-attach pod sa 3 ka letter sa strand delgado. So mao na to nga na-attach na. Ni-attach na ang tRNA sa kanang codon sa strand delgado. Mo-attach ang large ribosomal subunits. Remember, the large and small subunits, duha to sila nag-separate with the help of initiator factors. So, mobalik to siya attach ang kadtong mga initiator factors. For example, ang kadtong tRNA na attach to siya sa strand delgado with the help of the initiator factor IF2. Mao na siya nga ang initiator factor ni attach ang tRNA sa strand delgado niya. Ang small ribosomal subunits ni separate pod siya sa large ribosomal subunits with the help of initiator factors. Ang IF1 and IF3, so kadto siya nga mga initiator factors mo exit na to siya kay ni-attach naman ang large sa small sa subunits sa ribosomal. So mao na to ang initiator na place na ang tRNA, na-attach na siya sa codon ang codon sa tRNA sa codon sa strand delgado. Na-attach na ang codon sa tRNA sa codon sa strand delgado. So kadto siya nga site nga gibutangan sa tRNA, mao to siya stands with the letter P or peptidol chain nga ang iyang left side is the exit nga ang iyang right side is the entries of the amino acids stands in the letter A. APA na siya. So, sa elongations happens. Ang sunod nga tRNA which is nagdala-dala pod siya og amino acid. Always remember that tRNA magdala jud na siya og amino acid. Different kinds of amino acid na mo come up na makasynthesize siya na makahimo og protein. Mao na gitawag og protein synthesis. So moabot na ang ikaduha nga tRNA. So what if dili magka-match ang codon sa tRNA o sa codon sa strand delgado? So ang may tabuan na walay mahitabo na matching. Mawa to siyang tRNA magpaabot og lain nga mo-match sa codon sa strand delgado. So for example, na-match na naaman to'y site nga APA agad to siyang kinaunhan na tRNA naa siya sa P site. Inag moabot na tong usa ka tRNA na bag-o adto siya sa A, which is site sa sudlan sa tRNA, which nagdala og amino acid adto to siya. Nagkahuman ana agkadtong amino acid nga gidala sa tRNA na ikaduha na tRNA mo attach to siya sa amino acid nga gidala sa kinaunahan na tRNA. So mo attach na to siya, mahimo na og peptidyl chain. Magsumpot-sumpot na ang amino acid, so kadtong tRNA na kinaunahan mo exit to siya adto sa niya mo adto sa A nya mopuli ang ikaduha na tRNA. So mao na to ang process ana is magbalik-balik ra jud na siya sa elongation hangtod mahuman ang tulo ka subunit codon sa strand delgado. So sunod is termination. Magbalik-balik naman to siya hangtod ang tRNA mo exit niya ang amino acid, mag-chain na siya adto sa peptidyl chain. Kadtong site sa P, mag-chain na siya ang mahitabo ana mahutdan na siya og codon. Aw naay mahabilin na tulo. Ang mahabilin na tulo, ang kinalasan wala na siya ma-match sa codon sa tRNA. So ang mahitabo ana naay musod nga releasing factor. So kana siyang releasing factor, mao na siya maka-trigger nga agkadtong mga amino acid na nagdalag chain na ma-release na siya sa tRNA. So ma-release na siya, mauna na ang termination happens. So kadtong mga chain nga na-release katong mga amino acid na nag-chain na siya, mauna to ang i-synthesize para makahimo og protein. So maura to.
Change in the DNA molecules can cause mutations. One base pair is exchanged for another in the DNA molecule. One or more base pair inserted in the DNA molecule. One or more base pairs are deleted in the DNA molecule. There is a rearrangement of sections in the DNA molecule. There is an exchange of DNA region with another DNA molecule. That's recombination. So some mutations are harmful, some beneficial, and some are neutral. So what makes it harmful? Harmful mutation may cause genetic disorders or cancer. A genetic disorder is a disease caused by a mutation in one or more or few genes. It is generally caused by mutations in genes that regulate the cell cycle. Because of the mutation, cells will damage, DNA are allowed to divide without limits. I've read an article about a breast cancer woman in the America, and she has a second and third opinion already because she wasn't satisfied at first, because the first thought her that it was just about the lumps in her breast is just a lumps clog that's a result of after pregnancy, but she wasn't satisfied in it, so she had a second and third opinion of an OB/GYN. She have visited some far away hospitals from her town. So what she did is, in her second, she wasn't still satisfied, because same goes by as the first one, that it wasn't it was just a result of after pregnancy. So in the third one, it refers to have a genetic testing. I don't know if that is present here in the Philippines, but in America they have a genetic testing, because she have shared that in her history about her mother, that her mother had been a breast cancer. That's why they have recommend to have a genetic testing, and that's it. It was some sort of positive, or she has they have explained her that it was good that she had a genetic testing, because it was quite, there's a connection of her history. So I know we had already heard about this, that but she have written, or she have shared in her article, that there's a quote there that she says, knowledge is power. The more information you have, the better. Because she was always curious about her health, especially at her breast, because her mother died at a very young age because of breast cancer. So she wasn't satisfied with what the OB-GYN had given or had said or explained to her, so she had up to third opinion, and that's it. She had her unanswered question. So let's proceed to plasmids. Some plasmids encode for genes that enhance pathogenicity of a bacterium. E. coli plasmids encodes toxins produced and bacterial attachment to intestinal cells. Transposon are small pieces of DNA found in chromosomes and plasmids. They are able to direct synthesis of copies of themselves and become incorporated in the chromosomes, called jumping genes. Because of their ability to insert themselves into our chromosomes or change their locations. And lastly, transduction. Transfer of genes from a donor to a recipient by a bacteriophage. So this pictures is a very basic explanation of mutation. So the original copy and the mutant copy. So if it wasn't really 100% that he really replicate the original, so what makes it harmful if he replicates the abnormalities of what has the original? So that are some of the, so the very basic explanation of the mutation, that original and mutant. So that's it. Thank you.